And we're live. Welcome everyone to today's Science Thursday. Hugo Bowne-Anderson here from Coiled, although it's kind of Science Friday here because in Australia, we live in the future and we walk upside down. But please, welcome to today's Science Thursday with Matt McCormick, who I'll introduce in a second. Please take a second to introduce yourself in the chat. Let us know where you're watching from, what your interest in Dask is, what your interest in Jupyter is, what your interest in microscopy is, or all of these things would be great. But before I introduce Matt, I'd like to say, hey, Matt.
Hey, Hugo.
How are you, man?
It's good. It's good up here. How are you down there?
Pretty good. Pretty good. And I love doing these things bright and early in the morning, particularly when they're on topics so close to my heart and historically related to the work I used to do in biophysics and cell biology as well. And we have a connection there, which we'll chat about it at some point. I'll just remind everyone joining now to introduce yourself in the chat, where you're calling in from, what your interest is, and hit subscribe as well if you're into that type of thing and enjoy these things. But maybe I can just introduce you, Matt, and read your bio if that works. Or do you want to give a rundown of who you are?
Go ahead. Why don't you introduce and I'll poke your brain on your background in microscopy a little bit.
Great.
Yeah.
And we have I've just seen in the chat, we have, hey, Brian Parbu from Connecticut. Hey, Brian. Brian is someone who I met many years ago at a conference or a hackathon in New York City. So it's great to see you here. And Connecticut is a part of my cell biology journey as well, as we may discover the wilds of New Haven. But so Matt McCormick, PhD, is a principal engineer on Kitware's medical computing team, located in Carborough, North Carolina. His experience spans multiple medical, biological, material science, and geospatial imaging applications. As a subject matter expert, Matt manages and makes technical contributions to scientific image analysis projects. He's been a principal investigator and co-investigator of several research grants from the NIH, led engagements with the US national laboratories, and has led various commercial projects providing advanced software for medical devices. How's that?
That's a lot, but
and it's
interesting.
It's dense and meaty. There's a lot of great stuff there. Maybe you can, as by way of introduction, tell us a bit about your work in relation to Python and Dask and Jupyter.
Yeah. So Python and Dask and Jupyter. So yeah, I've been using Python for a long time, quite a while, and it's something that I discovered, coming from that academic world of biomedical imaging is really where I started, and kind of MATLAB was kind of the dominant language in the field in that day, and sometimes there are many labs still holding on to it, but I discovered Python when it was Fernando Perez was making IPython, and Travis Oliphant was creating NumPy, and then Matplotlib, and those kind of were the tools together that gave you more flexibility than MATLAB. But then through the years of doing more and more different types of imaging, like microscopy imaging, and Jupyter has come into the space, and I've always really appreciated the ability in Python to explore data, so interact with data and understand the values, do some computation on it, and get that close feedback loop. So in doing processing and visualization, I've kind of focused on that more recently, and doing things in Jupyter with Python interfaces. So it's something I've been using for a long time, and it seems to be getting more and more popularity, especially as people start bringing into the space machine learning libraries too. So everyone, like many other folks, very happy to see deep learning libraries, PyTorch, TensorFlow, and the whole PyData stack happening and flourishing in the scientific computing space. So.
Absolutely. And we may have discussed this before, but I can remember the time when I started using Python, and I was using IPython notebooks and trying to wrangle data into those, and the ability to with pandas to read in a CSV was a game changer for me. At that time, though, I was working in Dresden, Germany, in a cell biology institute, Max Planck Institute for Cell Biology and Genetics, which is a place very dear to my heart. The memories are deep and fond. And you have a history with the MPI as well.
I visited last year. Just yeah, it is an amazing place, a wonderful place, and some amazing people there in the open science imaging space, doing incredible work there. And so yeah, it's really fascinating that you came to that space, and we both experienced the work there in the small world.
Yeah.
So what types of microscopy and what types of data were you working with when you're?
So I wasn't doing experiments myself. I was working on modeling and theory, but we were essentially doing fluorescent imaging of in vitro microtubules. So for those not in the know, these are kind of parts of the cytoskeleton, the railway track of cells, where proteins are transported along by other proteins and that type of stuff. They form the mitotic spindle. So we were growing them and studying them outside of cells, and modeling microtubule dynamics of growth and catastrophe in particular. Yeah. But there were lots of interesting stuff happening in that lab at the time. People were building optical tweezers, and there was a lot of fun stuff. So I'd love to know what your interest in Dask is, how Dask is useful, and then maybe we can get into your talk and start chatting about your slides. And while we do that, I'll actually share the GitHub repository in the chat.
Cool, great, yeah, thanks. So there is a GitHub repository with some Jupyter notebooks associated with what we're going to be covering today, and folks are welcome to follow along as they'd like to. But yeah, as you're saying, we have this really fascinating data, this biological data that is the building blocks that holds us together, and we're trying to understand. And we have these really wonderful, accessible tools in Python that we can interactively probe and quantify and understand the data. But I find Dask so appealing is that it gives us the potential, the ability to take our interactive analysis that we might be running on a small subset of that data and scale it with that small amount of effort. So that's kind of the dream, is to scale, especially as our data sets are scaling in microscopy. We've seen in the last few years different modalities coming through, like light sheet microscopy, that produce very large volumes of data for single volumes. Or if you have many different samples that you need to process, Dask is sort of the dream. And really coiled, the company that you're working there, really the dream to take an analysis that you're exploring and interacting with a small subset of that data, and then just scale it up to a very large set of data. That is the dream, and I think that dream is starting to come true with Dask. And for folks that maybe aren't at a research institution or a large national lab or facility that have access to a cluster, or even if they are, and they want something that's just a little easier to spin up something quickly without different administrative hurdles or logical hurdles. I think the cloud-based dynamic computing that Coiled offers is very exciting. So that's why I'm really excited for the scientific Python space, this PyData space, and really the potential of Dask to bring us into this scalable, parallel cloud computing dream that we have.
Fantastic. That's a great introduction to Dask and how it applies in your work. And thank you for the kind words about Coiled. And essentially that is a lot of what we're trying to do, right? To make sure that people who want to scale all of their data work and data flows to the cloud, for example, or HPC, are able to do so with a click of a button and work from where they want to work best, whether it's a Jupyter notebook or PyCharm, or any number of other interfaces. So Great. So I think maybe it's a good time to jump on in and start talking through your slides. I'll encourage anyone who's joining us on YouTube, please do ask any questions along the way, and we'll try to answer as many as possible.
Fantastic. Excuse me. So yeah, so I'd like to kind of go through this interactive space and microscopy and imaging, and then walk through maybe how we can scale that with Dask and how it works in the cloud. So specifically, kind of the basics of how we get quantitative insights into images through some of these scientific Python tools, and specifically a library that I work a lot on called the Insight Toolkit, how that applies and how that can be applied to look into our imaging data. And then when we're doing this interactive exploration of the data in Jupyter or otherwise, how we can just take that and scale it up easy, live the dream there, as they say, and using the tools of Dask and in Coiled, in the specific example with Coiled. And those are kind of the computing and the software side of things. But another important topic that's evolving along with this is how to store and transmit data that works well with these new technologies and these new systems. So briefly talk about how that fits into these new chunked compressed scientific data formats that are used on the web. And finally, I know some folks may not be experienced with Dask, but I'm sure a lot of the listeners are very experienced with Dask and much better Dask experts than I am. But there is a problem that I think has to be solved, especially when we start doing this cloud computing and working with data that's distributed all over the world, Dresden, or in Australia, or in the United States. And as we do open science and share knowledge and share data, how do we work with that in an effective way? So the distributed data frontier. And
I like that, Matt. And I just want to interject and build on that by saying there's a good question whether the majority of Dask users in 5 years, we even want to see Dask. And I mean like the long tail, right, of people doing this type of imaging. Perhaps we want it to be abstracted over so they don't need to even concern themselves with the distributed compute in the back end, right? Agreed.
And so where things seem to be going and where I spend a lot of my time outside the scientific space, and I'm sure you do too, is software where you just go into a web browser and it's an application in a web browser. You go to a web page whether it's YouTube or other commerce applications. And I think the same is happening too in our scientific analysis. So what is the interface of the future? It's probably just a web browser. And whether you're explicitly using Dask and it's running on these very large data sets or not, we'll have to see that in the future. But I think probably what may be happening is you're going to a web page and things are scaling and computing locally and across the web. So,
and of course, this idea of the web browser, this interface is something Project Jupyter, for example, made an early design choice along those lines, right?
And it's been evolving, and it's kind of been going more and more towards the web. So Jupyter Lab, which has really turned into a client-side web browser application. So that seems to be the direction with Jupyter. Would you agree?
Definitely.
So that future of the web is coming, and that futuristic people like Elon Musk. He was on SNL. Did you see his show?
I saw some of the clips. Yeah.
Okay.
I didn't watch the whole thing, but
there's some good clips. There's some bad clips, but there was one clip, and I kind of highlighted it here, where he is looking to the future, where he's exploring space and working on Mars. this futuristic setting. But they, what I thought was really interesting is they had a screenshot there with some terminal output that was pointing to the future and a lot of cool technologies that are relevant actually today, and I think relevant to solving some of these distributed data challenges that will bring us to this future of parallel computing across the world that we interface to the browser. So we can maybe explore how that might work in the latter parts of our discussion.
Fantastic.
So yeah, there's a link here at the bottom of a GitHub repository that has some of these notebooks. Folks can go there. There's a README on how to get started. And
I've also posted that in the chat from the coiled account as well.
Excellent. So maybe taking a step back and how do we do some interactive computing and get some insights into our images? We can start with an example that we will iterate on and go through in our discussion maybe. So grab some data, some open data set, and this is a mouse blastocyst developing mouse embryo and some confocal microscopes. And first we want to really see our data. So there are tools to visualize the data in Jupyter. So the library that we have is called ITK, the Insight Toolkit. So you can import that, read the data from TIFF files if it's 2D or 3D data. And there's another package called the itkwidgets for working in Jupyter, and that lets us explore the data and probe the data and see what's happening with the data.
And you can do all of this in Jupyter Notebooks.
These can all happen in Jupyter Notebooks. And what's really exciting about it is that it's conducive to this remote data future where the data might be local on your system, or it might be in Australia, which is far away from me.
Yeah.
Or it might be in the United States for you. But you should have the same experience. So it's pretty exciting, fairly exciting. I don't know, what were the tools that you used when you were working on the...
I mean, because I was at in Dresden, Fiji was one of the big ones that people were using, and ImageJ.
Cool. Yeah, and standards, awesome tools.
Yeah.
So kind of what's exciting here is that you can work in 3D, which is really exciting, and work with these other tools. So after you see the data, maybe you saw that data set and it looked fairly noisy. So take an example, we might want to remove some of the noise. So this is how we could take that data and apply a number of filters in a processing pipeline, some simple filters, and remove the noise and examine the output. So this is what this library that we work on does. It helps, in a basic level, helps to explore and understand the data that we work with. So get insights into the images that we're working with. And this project is older than I am, although I am very old. It goes back, it started not in microscopy. It actually kind of has an really interesting history. The history is involved in medical imaging, and the first big project to following, we had the Human Genome Project around the turn of the millennium that tried to characterize all the genes in our bodies. Then this was a project with medical imaging to image an entire human body, two people actually, a visible female, visible male, and they took imaging data from the entire body, but they needed a way to process and understand that data, right? So we need software tools in addition to data, and so this project has developed over the past 20 years, and then it's an open source project, an open science project that is a collaborative effort with contributors from folks all around the world. It's relatively mature now with over 1.7 million lines of code, and it's continuing to evolve and brought to new challenges and new problems. So a lot of these new imaging technologies have some very large data sets. So one example is light sheet imaging. One example is serial blockface scanning electron microscopy. And in these cases, for some of these volumes where it may be 2 terabytes in size, so much larger than will fit in memory on any modern system. But still, we want to be able to characterize down to the level of what are the exact shapes and characteristics of capillaries in a mouse lung, for example. Ideally on the entire data set. And so how do we do this? How do we scale? Or how do we scale with large images that might come from some very powerful imaging systems we have? So these are from these light source, so very high energy X-ray systems that can create 3D tomograms of materials or biologic materials or biological cells at a very high resolution and creates very large amounts of data. But again, how do we actually get information from this data? Scaling is really the challenge. So some of the tools, the algorithms, and the methods available that are provided by ITK and other open source libraries are filtering, so processing. And this is pre-processing things to remove noise or artifacts that are natural, that are specific to a specific imaging method. And also to help do operations, higher level operations like segmentation. So identifying what are the different structures in an image. You get this big set of data, big NumPy array that has numbers. Well, we want to go from numbers to identification, like this is a cell, or this is the white matter in the brain. And so we need quantitative tools to help us do that.
And although this is very intuitive for humans to do on like an image by image basis, if you want to scale segmentation, it's actually, if you've got a bunch of circles and filled in regions that are, different pixels than others, maybe it's straightforward, but it actually is nowhere near as intuitive or straightforward as you would. I mean, as I mentioned before, I was working microtubule dynamics. So we had images of a whole bunch of little rods that may have been crossing it. So do you want to just get the cylinders, or if 2 are crossing, how do you treat that? Are those 2 different segments? And it's highly nuanced question, right?
And there's definitely a lot involved. So really, to understand it, you have identifying what are the tubules, and then maybe you want to do different analysis that involves maybe putting into a graph structure. Maybe you did that and doing some different metrics on the graph structure. So there are many different tools that need to be brought to the table. So ITK, the Insight Toolkit for pre-processing, is definitely one of these tools. You'll often use for segmentation a lot of the machine learning libraries for convolutional neural networks. You bring that in as a stage, and then you might do some post-processing on the output to modify the results of that convolutional neural network, and then maybe do something like put it into a graph structure and simplify it further. And what's so exciting about Python is that it allows you to bring all these tools together in such a constructive way. And I think what's so exciting about Dask is that you can use all those things together in a scalable way. So,
and it's easy to forget how really cutting edge and how new these tools are. My postdoc advisor, some of his earlier papers in the methods section talked about when he was measuring the length of certain polymers. The method section talked about like the type of VHS tape that he put, like that he used, and then printed it out, and the type of ruler he used to measure, you know, the length of certain microtubules and that type of stuff, right? So that was only a generation ago.
Yeah, things are evolving quickly, and at the same time, the other challenge we have with these tools is to keep that data around, keep that analysis around, and to be able to so that we can make progress. We have to be able to build on each other's work, and a lot of these tools and the efforts, the open source efforts, the efforts to preserve the data to make things sustainable and to keep working is really critical too. You know, so preserving that data, sending that data around is a challenge, and then these analysis, you know, so that a new grad student doesn't have to completely build the entire world from scratch. That's a new challenge too. I know a lot of people in microscopy, sometimes they have to, I'm always extremely impressed by the labs and the people that can go and build a microscope, one of these amazing microscopes from the ground up. It's incredible. But I would love to take advantage of their work and be able to analyze some of the data that they generate, and get bring it from their lab into the rest of the world, so we can learn from it too.
Awesome.
So then, you know, and another high-level operation that the Insight Toolkit helps with is registration. Did you do any registration, or do you know what registration is, Hugo?
Yeah, I do, but maybe it's worth before we start jumping into more Dasky stuff to let our viewers know.
Yeah, so it's another copyright situation is where you collect data at different time points, or you have different modalities and you want to compare them. So here in this figure, we have two different modalities, and we might want to compare what is the result of how does the tissue present with these different energies in these different modalities. And so you have to align them in space. So that is registration, and that is one of the things that ITK can help do, is you line in different space, and then you can start to do additional quantification after the data has been aligned. And so, you know, what are the things that's unique about ITK, or that it does very well after using it for many years as a library, as a framework, is that it is all the operations are accounting for this world space, this spatial framework. So the data is living in a coordinate system, a world coordinate system, and we have the location of a pixel that has a specific location in physical space, which is, of course, very important with microscopy images, because for most modalities, there is anisotropic spacing. So in the z-plane direction, it's usually coarser than in the in-plane direction. And it's important when we want to do registration and align things and account for things. So we've been adding more support for xarray, and xarray is a labeled array, so it allows you to label different axes, which is often a source of confusion. And you know, when you're transmitting information, you know, what are the different axes? So it's very helpful in that sense. But also, when we're using it in ITK, it allows us to identify the coordinates of different pixels in the image and where they live in space. So then images is part of it, but also there's these other data structures that are also important too, like representations of spatial transformations. So if you want to start to explore some of these insights from your images, there's a number of resources here. This This is linked in the presentation. And in the notes, there's quick start guides, there's Sphinx examples. There are a few plugins for Napari. I think we're getting more. I know Napari is powered by Dask, and it's a very powerful tool if you're trying to do visualization with a desktop application. So you can do registration. There's a large book, discussion online. There are, if you want to support the project, there's it's a non-focused sponsored project, so you can support the project in that way. Or if you need commercial support, the company that I work for provides commercial support too. So, my dream, and I think maybe you're helpful in making my dream come true, Hugo, is taking these things and then just saying, press an easy button and run them on my own cluster in the cloud. And that dream is possible.
And a dream is our mission.
Thank you.
Right?
Thank you for helping making that happen. It seems like it is progressing quite quickly. And so thank you for helping make that happen. And thank you to the community who's really helping make that happen, the Dask community, a wonderful community that has really matured and developed. So Dask, right, is this technology and tool that allows us to scale our Python-based pipeline, everything that may be in it. And it does that with these data structures. And then Coiled, right, is this cloud service that we can hook into to create a cluster. And how would that work with this type of analysis that we just went through, this example analysis? How do we scale and parallelize onto a large data set? So this data set here, it's actually not that large. If you have a reasonable system, it's not necessary to use a big cluster, but you can imagine get a much larger image that you might want to run it in parallel on a cluster. So the first thing that you do is maybe chunk up the image into different pieces. So very simple, easy data parallelism. And these n-dimensional images are very conducive to that. And so you take that image, and if you create a NumPy array from it, and then we can use the Dask array data structure that I'm sure you're familiar with, Hugo, but maybe other folks might not be aware. And that takes the block of data, and you specify how you want to chunk it up. And the result is a chunked data structure representing the image. And then we can specify a function that we run on every one of these chunks in parallel. So we take that processing pipeline that we had before and put it into a function. And then we call map blocks. So we say, run that function on all these different blocks of the image, and then Dask will go out and it will plan how to do that on all these images. Specifically with ITK, there's a few things to be aware of. So one is that Dask does allow you to do parallelism even on a single system, if you have a multiple core system. But ITK, it does its own threading for almost all of its operations already. So you can actually get too much parallelism, and that's called oversubscription, a technical term for computing term. When you are telling the computer to do too many things at one time, it kind of gets confused and it can slow down. So if you were trying to do parallelism with Dask locally, you could say ITK set in threads. This is actually Matt Rockland's suggestion. Call it that way. We have that now. And that will, if you're doing parallelism looking, you might want to call that inside your function. And another item to be aware of to make this parallelism happen is that there's some constraints, is that you have to import ITK inside this function. And this is sort of technical detail. But as you encounter different libraries, and we've made some improvements with ITK, and there's more improvements to be made. But what is happening, I think that a lot of folks don't understand, and now I understand after working with Dask for a little while, is that Dask is really serializing your function, so the operation, the Python code that you have, and the data, so your inputs and your outputs, it is serialize them in Python. That's pickling, put it into a jar, and that has to happen in a nice way. So some scientific libraries may require improvements to make that happen, and so one of the current caveats that we're improving with ITK is that you have to do the import, so Python can find where this operation comes from when it passes this function around.
The other thing to note, I think one of the things I love about Dask, and people who are regular attendees will have heard me say this before, how Dask is as invisible as it needs to be, right? So if you go back to the previous slide, we'll see firstly that the API for Dask is incredibly similar to NumPy, and in fact, not only do we treat it like NumPy arrays, but in the back end, it consists of a set of NumPy arrays as well. So our mental model of what we think will be happening computationally is actually what's happening. So this speaks to, I mean, there were certain technical goals of Dask development. Matt said when they initially developed it, there was also a social goal, which was to invent nothing. So to as much as possible, to give people the APIs and the structures they already know and love.
I think it makes a lot of sense, and I think we're trying to do the same with ITK to some degree. I think there's no need to create something new if you don't have to, and being familiar is a big advantage. So adding when you do read in an image, this is an ITK data structure, but just like Dask arrays, these data structures try to be familiar. Having attributes like shape on them is helpful, and that's an evolving convergence that is happening.
Great. And Matt, I just wanted to say we've got just over 20 minutes left, just to calibrate with respect to time.
Okay, great, thanks. So the other thing to realize about Dask is that you have to call compute to actually make things happen, and this is something that is related to lazy computation. But this is an advantage in that if we have very large data sets, that lazy computation allows us to set up that computation and then run it at a different stage, so that we can not load everything into memory and process data sets larger than memory. And as another detail, we have for these operations where the impact of the computation might require a context of data, you can use another function called map overlap, and that will take in and run the function on a larger set of data, that context that's needed in the image to denoise that local data, and then remove the excess outside. And how do we scale that to the cloud? Well, here is the easy button. So Coiled, allows us to press that easy button, essentially set up a cluster, and it will guide you through the process of logging in to a simple website, again, kind of a website, and provide this the dashboard. And after you do that, you just run the exact same code, and it will run on the cluster. The one kind of caveat that folks coming into who are new to Dask and Coiled may be aware of, is important to be aware of, is that you have to have the same code, the same software packages running everywhere, and Coiled does provide a very convenient way to make that happen in taking a con environment or a pip requirement.txt and building that into a Docker image, and that's used the same. You can use that same environment locally and in all the workers on the cluster.
Yeah, and this, of course, is one of the biggest pain points that we've seen a lot of people trying to scale their Dask clusters and workflows to the cloud, making sure that, so that was one of the first things we wanted to do, was making sure that we had people's software environments locally and on each worker sorted out.
I think if you're not using Coil, that can be a real problem, but it makes it much easier in my experience with that. So there's a lot of resources out there. The Dask documentation is good. The Coil documentation is good. Dask image is a specific package that's useful for working with Dask. There's also some more blog posts and tutorials that people may find interesting. So before we run out of time and have time for questions, I'd also like kind of look into quickly, how do we work with data that works well with this structure of working on the cloud with Dask. So there's these new formats coming out of the Python community and also the ImageJ community, sort of convergent evolution, I would say, or intelligent design. Maybe if you ask the authors, they might say that. But these different file formats are conducive for working in the web and doing this parallel computation on large data sets. Compressed data, so that their characteristics are, you have this group hierarchies, you have this web JSON format for the metadata, and these chunked n-dimensional binary tensors or arrays that we're used to working with, and very good compression. And recently, the community is starting to standardize on this format with image models for working with microscopy images, and that is this next generation file format. And the open microscopy environment folks have been kind of leading that effort. So this is useful. And so what does this mean? Basically, it's the same as what you had before, is you can use this zarr format, but it loads in the data already in this chunked way, and you can load in the data from web resources and write to it in parallel with Dask. So great connections there. And folks are interested, and I encourage them to look at some of these resources, the forums, and the documentation, and some tutorials. But now into kind of unsolved problems in the future, working with Coiled and Dask. So it's awesome that it makes the easy button for creating a cluster and doing the compute. But one of the challenges that I've seen, and maybe you've seen this too, is what do you do with the data? How do you get the data there? And how do you do that effectively? And this is the nice new Coiled. I got this from the nice new Coiled website, which is beautiful. I
love it. And maybe could you talk us through that schematic a bit more?
So yeah, so here I am on my notebook. That's
looking great.
Looking great. Maybe it'll work out a little bit more, but then I'm using, here's my nice easy button that you work hard on and sweat over, but a lot of complicated things that I don't have to care about. Yeah, or other people don't have to care about. And then here's our nice cluster that's providing these compute resources for us. And so that's running the Dask cluster. That's awesome. But kind of the question is, where do you put the data? How do you get the data there? And this is on AWS in practice, right?
Mm-hmm.
And sending data in and out of AWS. What if I'm not on AWS? What if I worry about ingress and egress charges on AWS? How do I get it there? So when Elon was working on Mars, he used these nice technologies, many I love, like TMUX and Linux and ARM processors. And one of the technologies—
I love that you zoomed into the code to see what was happening there.
I was fascinated, very fascinated to see what he's using. And one of the technologies he's using lately is IPFS, which is, I found fascinating because I've been looking to, and many folks in the community have been looking into too. And what is IPFS? IPFS is the Interplanetary File System.
Wow.
And that sounds very exciting, and it is very
Sounds
exciting.
cosmic.
It is cosmic. It is out of this world, and it's a protocol and peer-to-peer network for storing and sharing data in a distributed file system. And how does it work? What's unique about it? It uses content addressing. So think hashes, we see them all the time, to identify data instead of locations. So instead of going HTTP ://coil.io, I go to Baffy12345. And how can this help us with this problem? And what is this problem that we have? So if you're there in Australia and you're trying to use the coil cluster, which is in the United States, you're sending that. You have to send, if you have data locally, you're gonna have to send that data all the way across the world every time that it's needed, right? And that's bad. That's bad for performance, and maybe bad for reliability. So what would be better is that if you had the data and you're using open science data, as you're a open science guy, and we have other people in the world that are sharing and having this data locally too. When you say compute, it goes out and looks for that hash and it finds it wherever it can find it, maybe from any of our friends in the scientific computing microscopy world.
And I love that several of the people here are people I've worked with in the past, and people from my time in Dresden, but they're on different continents now.
People get around this world in a great way. I also have Matt up in
where is he?
Mars. Yeah, he's on Mars. I know he's a
I wouldn't be surprised.
Digital nomad, and I wouldn't be surprised if he'd be on Mars.
Absolutely.
He could be there. But what can happen with this distributed file system is it can get the data from the closest location when it first fetches the data. Not Matt, even not you, but maybe like a data center that's local that provides services to host this data, easy to host the data. And what would be even better is if once it got the data the first time, it's used locally and it passes it around to the workers in the cluster, and it doesn't have to keep going out. So I think this would be a big optimization to what's happening now in the file system. And the cool thing, the amazing thing, is that we have this new data format, this zarr, which it works in cloud resources in AWS S3, or it can work on local file system. And there's a very simple mapping of a system of files and folders that zarr is made up of. And also this IPFS supports systems of files and folders. And it also supports chunk data in kind of in the same way. So there's a very simple mapping between these two things. So kind of giving these content identifiers to these chunks in a nice way. And so some people in the community are exploring this. A fellow created a package called Ipfs Spec. And so you can use that in the same way with zarr and Dask arrays. And so right now, that's read-only. So it gets us, advances us, evolves us into this place where we can get the data locally, and you wouldn't have to put all your work into sending it from Australia and kicking it all the way over the ocean. But we can also make a few more steps, and I think it'd be really interesting. The people in the community, people at Coiled, and some of the brilliant folks like James and other people in the community could make this happen. And what would be required to make that happen is, instead of just grabbing the data dynamically, it'd be starting up a node on each one of the workers if there's not already a node to share the data, and then directly connect them with each other. So instead of having, there's this distributed hash table that's required to transverse if you don't know where the data is in your connections, but we could directly connect these swarms together. So that's kind of one idea to help that might be useful to bring us into a web transparent future, parallel computing everywhere future. And so this is exciting, really exciting and new. I think it's personally, it's one of the most exciting things that I've come across. It seems to, it's very technologies that change the world, and they come across, I think, every decade or so. One was scientific Python in my experience, and I think this is maybe the next transformative technology. So I think it's an interesting technology to explore, and it's been around for maybe 5 or 6 years, but it's finally getting to that point, kind of where Python finally got, where it has the potential to really take off. So I encourage everyone to investigate it and see what you can do with it. But I'm interested to see what we can do with it in the Dask community in the future. So that's all I had, Hugo.
That was fascinating. I have so many thoughts and questions. I think maybe we can, spend 5 or so minutes chatting around a few things and then wrap up. I think there are a lot of interesting details there. One and a very nice high-level vision of where the space has gotten to and where it's moving. What I'd like a bit more insight into is how it changes how people do science, how it changes the working life of a research scientist or someone working on this type of microscopy. Does it shift timescales of their work and how they get in flow with their work? Or
I think as we go into the future, some of these new technologies, one of the essential principles is that the data must be local to the compute, which must be local to, in most cases, it's like local to the microscope. So probably when you, a decade ago, and probably in most labs today, if you try to do anything with a microscope, you're doing it at the microscope, and then you get the data and you're doing the compute there on a workstation. And that is kind of limiting in what compute resources you get access to, and who can get access to this data, and kind of what the impact of the data. So as we start making things available on the web and work well on the web, more people are going to get access to that data, and the people who do have access can do more with it because they'll have access to more compute resources. So I think that is really exciting too.
It's incredibly exciting. I'm interested clearly in the role of open source. I suppose I'm wondering your thoughts on ITK and the fact that it is open source and how that has helped it get to where it is today, as opposed to other paradigms of software licensing.
I think the project has gotten to where it is today, I think, because it's open source, mostly because of the community and the people who have contributed. So what is so compelling I find about open source is you have these brilliant people all around the world who maybe wouldn't get together, and they self-select and cherry picking and get to work with each other and create beautiful, wonderful things. And also, you get to meet fascinating people. So I just, open source, it produces wonderful things, but it is also a great way to connect with people, be in Australia or otherwise. So I think ITK has really benefited from many contributors. There's been some fantastic contributors there. Richard Bear comes to mind. He's from Australia too. He's made some amazing contributions, and other folks who contribute to the project. And I think that, for me, that's one of the most exciting parts of open source is the people who do come together to contribute.
Absolutely. And my understanding is Kitware develops a lot of open source software across a bunch of different fields.
Yes, yeah. So you can take a look at the Kitware website. So one of the projects we support is ITK. Another project you may be familiar with is a visualization toolkit. And that focuses on visualization, scientific visualization. Another project you may be familiar with, as you see terminal code scrolling through the screen when you're compiling anything, is a project called CMake, which is a build system that's used. It started with scientific tools. Now it's used kind of across the programming industry for C and C++ build systems. So these things, they have a large impact, and I think that's makes them so exciting, is really the adoption and the use they can have.
Fantastic. And I've also, so to wrap up, I'd actually love to, for people who want to know a bit more about ITK, definitely check out the repo that we shared in the chat, and check out ITK's website. I also noticed that if people are really interested, Kitware's hiring.
Kitware is hiring. We are definitely looking for those folks around the world who love open source and love to do imaging and visualization and web-based computing. So if you're interested, go to kitware.com/jobs, I think.
Yeah, /careers.
/careers. So thank you.
Yep. And itk.org has a link right at the front on it as well.
Perfect. Yes.
And of course, wonderful that the ITK logo has a snake as well.
Yes. Yes, that is a snake. So that's part of the medical history. That is the rod of Yes. So if you're familiar with kind of the logo of doctors.
That was actually part of the inspiration behind Coiled and our logo as well. I mean, it's a highly abstract vision of it, but you can almost see it, right?
I can see it a bit. You know, Python is cool. We love
absolutely.
We love Python. It's part of the programming language, but
yeah.
And I do love the new logo that you guys have. So looking more and more stylish.
Yeah, and we all love it, which is against all odds, I feel.
Logos are very controversial. I don't know how you made that happen, but you're
seriously. No, blood, sweat, and tears, right? All of the above, because aesthetics is something that it's very difficult to communicate around as well. You know.
Yes.
All right, Matt. Well, we're at time. If people want to check out the repo, please jump in. If you're interested in jobs, check out kitware.com/careers. Is there anything else that you'd encourage people to check out? That's pretty comprehensive, but
I think that's great. Thank you so much, Hugo, for the opportunity to talk with you and taking the time for getting up early this morning. I appreciate it.
Absolutely. It's always a pleasure to chat with you, Matt, and I love bringing these live streams to our community as well. Awesome. All right. Well, thanks everyone, and join us for next month's live stream. And